Yeah, this is Dr. Muthu, Assistant Professor from SR University, Warangal. So, in this video, we are going to see everything a person needs to know about the small molecules. So, what is mean by small molecules? So, in an easy way, we can say that as a drug molecules, so or inhibitors, or what are the molecules which is smaller in size. So in biological term, we have two different types of molecules. One is large molecules, or we can call it as macromolecules, which is also known as proteins. And we have small molecules, which is also known as drugs or inhibitors. So compounds, molecules, so all this comes under the same category. So today's video, we are going to work on how to generate a three-dimensional structure of the molecules and how to draw the structure of drug molecules and what are the easy and freely available tools which is available. We are going to see some of the tools and we are going to work on some tools to generate the 3D structure of the molecules and how we can retrieve after the structure generation, how we can retrieve and how we can process those molecules for further applications. So everything we are going to see in this, and after that we are also going to see what are the databases available to get the instant structure of small molecules. In small molecules we have a different file formats: mol2, sdf, like PDB, and also smiles. So like that, we have a several kind of biomolecule file formats. So we are going to see one by one today's lecture. So one of the well-known application to draw the small molecule, as you know, that ChemDraw, and we have a software called MolView, so which is freely available, and it's a web server. So we no need to install any of the tools in the system. We can draw it in the online itself, and we can easily download the structure of molecules, and easily we can visualize and we can tweak. If we want to tweak any specific functional group, we can easily delete and reattach the newly generated functional groups. So everything can be done in the particular Malview. So when you opened the Malview website, you will have an option to choose either new app or the old app. So I'll show you both. So what are the drawbacks, what are the complications available in the old app as well as new app. So we'll go to old app initially, so continue to old app. So here you will have the two different interfaces. Okay, this is interface where we can get the three-dimensional structure, we can rotate and we can see, and this particular area we can draw the structures. So whatever the structures we want, here also zoom in zoom out will be there. So here you have the delete option. If you click this clear all, so that entire structure will be cleared from the screen. And this is the eraser button. So if you want to clear a specific thing, we can do this like this. So if you want to clear the specific things, we can clear using like this. And this one, if you want to undo it, so we have did some wrong structure, and in place of here we did it here. So if you want to replace it, sorry, undo it, so there is a button called undo as well as redo. And if you want to move, so like this also can be possible. If you want to delete a specific portion from the structure, so there are two tools are available, and bonds and then these things. And once you generated the two-dimensional structure, so we have to click 2D to 3D. So whatever the structures we draw as a 2D this side, it will be converted to the, yeah, optimized three-dimensional format. So here we can see something like this. A three-dimensional structure we can see, right? So now we are going to generate the structure of aspirin. So for example track, I am taking aspirin is a well-known chemical. So how we can generate the structure of aspirin here? So I am taking the 2D structure of aspirin, and here we are taking the Malview structure. So how we can generate it, right? So here there is an inbuilt benzene ring is available. So we no need to draw the benzene like this one by one like this. So there are inbuilt benzene ring included with all the bonds corrected. It is available. So we can directly take the benzene ring to be here. So if we want to rotate or any of the benzene rings or any of the bonds, so while placing itself we have to rotate. Here you can see, so while placing the benzene ring or while placing any of the bonds, we can easily rotate like this, right? So we want to be like this, so we have rotated just like this. And so this particular side we have individual elements. So here very fundamental elements are mentioned here. If you want to apply any chemical from the periodic table, there is an option here. So you can see all the elements from the periodic table. So you can select whatever the elements as per your structural requirements. So just now I have placed the benzene ring. So then we have a single bond. Here we have a single bond, and we have a double bond. So we need double bond we have to put. So either we can do like this, or you can put single bond, and if you click on the single bond, so it will be become double bond. If you click it again, it will be become again single bond. So we need double bond, right? So we can do the double bond, and we need the oxygen molecule. So we are placing it here, oxygen, and we can take another single bond. So oxygen, then we are going to implement the hydrogen. And when coming to this side, so I'm just adjusting the structure to this side, so it will be easy to draw that. So then we have a single bond. So then we have an oxygen atom. So then single bond and another single bond. So we need to put double bond oxygen here. So the structure of aspirin has been ready. So once we generated the structure, so if all of the bonds are correct, so we can implement 2D to 3D conversion. Here you can see, here we can easily see the three-dimensional structure of aspirin. So whatever this the like cement color structures, so that will be denoted as the carbon, and this yellow color, sorry, red color will be denoted as oxygen. If you want to include the nitrogen, we can include. So nitrogen we have included for example. So I just converted to 3D. Nitrogen will be shown in the color of blue. So you can see here. Here I only added N, but I didn't added the hydrogen atom. So nitrogen have a 3 NH3. It should be there. So this hydrogens are automatically added here. So that is a one main advantages of this particular website. So once we generated the three-dimensional structure of the molecule, here you have the option called tools. If you want to generate the three-dimensional image, if you click here 3D model image, that image will be downloaded, so that you can use it for the any another purposes. So even this image quality also very good, very much nice. So whatever the direction, whatever the rotation of in the three-dimensional space, if and you can rotate, and you can have a snapshot. For example, if your study focusing on altering the functional group, so if you want to alter this specific particular part, so let's say you are asked, so like this. So again 2D to 3D. It should not be there. Wait. Let's say I'm adding sulfur here. So I want to highlight the addition of sulfur. So we can rotate like this, and we can have a image tools, 3D model image. So like this we can take it. Yeah. Let me. So the another form, how we can take the 3D structures. So you can go to tools and mol file. So mol file is a specific format which can be. So I'm just putting it here to the new folder. Yeah. So here you can see this is a mol file of our molecule. I'm just renaming it to molecule. So here you can check the properties. Go to properties. Yeah, here you can see the structure of the particular file mol.mol. There are many file formats available for a particular drug molecule. So I'll show you how we can convert from one file format to the another file format. So we can see one by one. I'm going to the website of several databases available to retrieve the chemical structure. The well-known website called PubChem, which is comes under NIH. So if we want to download any small molecule drugs, we can search. For example, if I want to download aspirin structure, I can easily search aspirin, and I can go inside, and here there are many file format. You can see 2D and 3D. So mostly we will use these two structures, and we will sometimes use this notation called smiles or inch key. So this particular thing. So smiles is a primary format of the any chemicals. It will be in the alphabetical and this thing. So here you can see the option called download, and if you want to download the structure in two-dimensional format, you can download, or else if you want to download in three-dimensional format, here also you can download. So here, this is SDF, structural defined format. So that is also one of the file format which is available to download the three-dimensional structure of the molecules, right? Yeah, that's the same molecule I have downloaded in the form of SDF, right? So now we are going to see the another database called drug bank. So here this particular database contains the drug molecules which is in the form of investigational stage and approval stage, and also the experimental stage. You can find all these drugs, for example tramantinib, and also you can find a different file formats to download like pdb, sdf, and mol file formats available to download. And the best feature of this particular website, we can identify many properties of the drugs including the ADMET properties. Admet properties is nothing but what are the pharmacokinetics and dynamics properties of a particular drugs. Here you can see this particular drugs, what are the clinical trials. So it is under clinical trials for the phase 5, phase 4. With the 5 clinical trials experiments are currently going on, and phase 3 14, phase 2 158. There are different stages of clinical trials we have. So all the informations we can get it from the website called drug bank. And pharmacological, what is the use of this particular drug and what are the diseases it is been working with. So everything will comes under the database called drug bank. So what we need the download option. So here you can see the drug image, and here you can have the options to download mol file, as we have in the pubchem, sorry, mol view also. Then we have sdf 2D and 3D sdf, then pdb protein data bank, which is also one of the format for small molecules. Then smiles, it's a primary notation. Then inchiki. So these are the different file formats available in the drug bank database. So let's say if you want to perform a further experiment, which is also called as like molecular docking calculations. So what are the file formats required and how we can convert? So that we're going to see now. There is an important tool called Open Babel. So I think many of you have heard about this one, Open Babel. So this particular software will helps to convert from one particular format to the another particular format. So with whatever the modifications you want, you can do that. So in this also, there are two options available. One, you can do it in the website itself, like web server, it's online server. Sometimes it may have lagging. And the another options, you can download the particular software and install it in your local computer, and you can proceed with the conversion. These are the two options. So this open table is available for Windows. Yeah, here. Sorry first. Here you can download this particular software and you can install it. Yeah. So once you have installed the folder, so you may get the folder like this, and here OBGUI is nothing but Open Babel graphical user interface. So it will be like this. So as I said earlier, so there are two different options are available. So this Open Babel is an online website. You can have a limited options, and this is graphical user interface. Like it's a kind of same thing. So both are looks like same. And there are one more options available. It's a terminal interface. So you can have a terminal interface like this. OBabel. So which is nothing but Open Babel. Here you can see when I type OBabel, so it is asking the missing file. So what it is that? So OBabel, the first thing I is nothing but input types. What is the file types for the input molecule? Let's say as I said earlier, if you want to convert from sdf to mol file, or mol file to sdf, mol file to mol2, all smiles, or whatever the formats. So what is the file type for the input? So that is needed, and we have to input the file, particular input file. So this is I, and input file type, and the particular file, input file. Then hyphen small o. So we have to be very careful, that is small o, and output file type. Let's say if you want to convert from sdf to mol, so here you have to write obable hyphen I is nothing but input file. Let's say I want to convert from sdf to mol, so I have to type isdf. Then let's say compound dot sdf space, then hyphen o, output type. So output type is nothing but I want to convert to the mol file, so mol. So isdf and hyphen imol. So then we have to type capital O. then file name. So let's say compound dot mol or mol2. Okay, mol. So here, whatever the output file format which is mentioned here, the same file format should be mentioned here. File name, it is optional. You can write as per your convenience. But the format, if you are entering mol here, you should enter mol here. You should not enter mol2. If you want to enter mol2 here, you can enter mol2 here also, right? So these are the several tweaks we can do in between. So if you want to convert this, let's say we have a 2D SDF file. If you want to convert it into 3D, so hyphen hyphen gen 3D. So like this. So in OpenBabel, we can optimize also or be minimize. So that option is also available if you want to energetically minimize the particular small molecule. So that particular option is also available. We can work on it. These are the force field. MMF of 94, MMF of 94S, and UFF universal force field. So these are the general force fields which is available in the open Babel. So here we will convert a small molecule here. Let's say these are the molecules I want to convert. So in a biological or bioinformatics experiment, the foremost care we have to take in care is, so whatever the folder we are creating, we have to create without space. So that is the important task. Or we should not use space in any of the locations, right? So let's say this is compound one. We should not type compound one. So we have to use this underscore or hyphen or whatever it is. So we should not use space in any of the locations, even with the molecule name, even with the folder name. Then only all the softwares will work perfectly, right? So let's say in GUI we will convert first. So this compound one I am dragging into here, and we want to convert. So what is the input file format? So compound one is in the form of mol, MOL. So we have to search or this MOL format, right? So we want to convert this small file into the, let's say, SDF, because it is already available. So just for the sake I am converting. So once you set the desired format, you can click convert. So the molecule is converted. Either you can copy paste this one, or while converting itself we can put the molecule name here if you want to output file mol.sdf. So like this we can make it, and we can click convert. The mol files will be generated automatically. So these things also we can do, or else we want to convert it in the terminal, because terminals will be used for the a bulk task. So in that GUI, we upload all the files together in a single file format. Let's say SDF file format. We can do that, or else, yeah, terminal is the easiest way. And if you want to provide, or if you want to convert the bulk compounds, we can easily convert with the help of terminal. Let's say before conversion we have to go to the particular directory. How we can go? We have to type cd is nothing but, if you want to go to any directory, we can type cd and we can paste the particular directory. It will be directed to the particular location. Let's say previously it was C program files and open babel. Now this particular thing has came to the directory of molecules which we entered. If you type ls, you can see that these are the two compounds which is already available, right? So let's say if you want to convert, let's say first file, if you take aspirin.sdf, these things we want to convert in the form of PDB. So how we can convert? So O Babel. So let's say I'm typing without entering all these commands. So here you can see O Babel I O as we discussed earlier. So these are the file formats available. So O Babel hyphen I aspirin.sdf. So the input file format is .sdf. SDF. So ISDF and aspirin.sdf we enter. Then hyphen O. So if you want to convert to PDB, so PDB format, so aspirin.pdb, right? So we want to convert from SDF to PDB. So here we have missed the argument hyphen capital O. So that is the important one. OPDB is nothing but the output format it will decide, and hyphen capital O. So that will decide the output file name if you want to convert. So here it is showing one molecule converted. Here you can see we can open in the form of this thing. Here you can see SDF file. So this is structural defined file format. So here you can see we go to the properties. So since I have installed the Rasmal, it is showing like that. So the format you can see here .pdb, right? That's also you can open. So these are the two different file formats. Aspirin .sdf. This is aspirin.pdb, right? So we have successfully converted from one particular file format to the another particular format. So this will really helps to found the molecular docking calculations, and if you want to perform molecular dynamic simulation, so whatever the file format is required, we can convert with the help of Open Babel. So all these things to know about the small molecules, how we can convert and how we can process the small molecules, and how to create a small molecules with the help of a 2D structure. So in most of the chemical laboratory, so they may draw the structure by hand, or they may attach a specific functional groups in place of sulfur, let's say F, and let's say Fe. So like this, they may change different modifications in the functional groups, and finally they may get the desired structure. And we can generate, let's say 10 structures we can generate, and computationally we can test against several target proteins with the help of studies like molecular docking and dynamic calculations. Then we can find out the foremost important structures. So these are the things. And we have another database called Campbell. So this is also the another database. So where we can get the small molecule structures, right? Yeah, let's say here also tromatinib or aspirin. So this also, yeah, one of the well-known database, and this is a very larger database which contains, it is like integrated database. This particular database you can see drugs as well as the small molecule targets. So and we can get the biological activity values of all these drugs. So here you can see bioactivity, how much inhibitors, KD, how much number of experiments it has found. So all these things we can see what are the target enzymes associated with a particular small molecules, and so everything we can see with the help of Campbell database, right? So in today's lecture we have seen how to generate a three-dimensional structure of a small molecule, how to convert from one file format to the another file format, and what are the basic tools available to generate the three-dimensional structure. And we have seen several databases where we can get the structures instantly. So all these things we have seen today.